Neurons and the nerve impulse. So, overview of the nervous system. If you recall from previous classes, the endocrine system and nervous system maintain internal coordination. Those are the main methods of communication in the body. So, the endocrine system communicates by means of chemical messengers known as hormones that are secreted into the blood and travel through the bloodstream to a location that's far away or at a distance from the organ that's initiating that secretion of those hormones. The nervous system utilizes neurons or nerve cells to send messages from cell to cell by electrical and chemical means. And this is going to occur in 3 steps. Now, the chemical means we're going to learn about are neurotransmitters. Neurotransmitters are chemical messengers that jump between the adjacent neurons, while the endocrine system uses hormones. But the 3 steps of those sending those messages down nerves would be: it receives stimuli from the external environment and transmits messages to the central nervous system. The central nervous system then processes the information and determines what type of response. And then the CNS issues commands to muscles and glands to carry out the response. There's 2 major anatomical subdivisions. The central nervous system contains the brain and spinal cord, while the peripheral nervous system would be all of the nerves and ganglia that kind of branch out from the central nervous system. Peripheral nervous system is functionally divided into sensory and motor divisions, each with somatic and visceral subdivisions. The sensory afferent division carries signals from receptors or sense organs to the central nervous system. So sensory, it's bringing in senses from all of your senses all over your body, bringing it back to the central nervous system, the brain or spinal cord. The somatic sensory division carries signals from receptors in the skin, muscles, bones, and joints. So if you've ever sat at the doctor's office and they took that little hammer right below your kneecap and pounded it and then you kicked out, they were testing a somatic sensory reflex. So we're going to learn a little bit more about that in this course, and we're going to talk more about the different types of senses and sensory receptors. But that's the main one there, is it's going to carry signals from the receptors in the skin, muscles, bones, and joints. Typically, a lot of things that are under voluntary control. So we can voluntarily move our muscles, which causes those bones and joints to move as well. Visceral sensory division carries signals from the viscera, like the heart, lungs, stomach, and urinary bladder. Those are typically under involuntary control. We cannot voluntarily tell our heart to beat a certain way, so that's involuntary. It's going to beat according to its own electrical conduction system. The motor or efferent division carries signals from the central nervous system to the effectors, glands and muscles that carry out the body's response. The somatic motor division carries signals to the skeletal muscles, which causes voluntary muscle contraction and autonomic reflexes or automatic reflexes. Visceral muscle division, autonomic nervous system, ANS, carries signals to glands, cardiac and smooth muscle. There's no voluntary control here, and responses are called visceral reflexes. In this division, we have the sympathetic and parasympathetic divisions. The parasympathetic division of the autonomic nervous system stimulates and prepares the body for action. Sometimes we call that fight or flight. So the fight or flight symptoms or system that's in place helps regulate and coordinate all of the actions that are going to help us either fight something or run away from something or have some major kind of burst of energy to be able to escape danger. Parasympathetic division has a calming effect on the body. I sometimes refer to this as the rest and digest. So under parasympathetic division, we would regulate sleep, we would regulate the digestive system, the formation of urine, all of those body processes that we do when we're not having to fight something or run away from something. We're going to learn more about those two divisions and what they entail in the next lecture. So here are the main divisions. We've got the central nervous system broken down in the brain and spinal cord. The peripheral nervous system has the sensory division and motor division. Sensory brings information to the central nervous system, while motor takes information from the central nervous system back to the glands or muscles that are going to perform an action. Within the sensory division, we have visceral and somatic. Somatic's going to be more so related to the muscles and bones and joints and skin, while visceral would be like the visceral organs that are under autonomic control, automatic control. The motor division is then further divided into the visceral motor division and somatic motor division. Very similarly, we're talking about the causing the movement of voluntary movements, so organs that are normally under voluntary control, like our skeletal muscles, or the movement of visceral organs, and those are normally not under voluntary control. Under the visceral motor division, we can break that down further into the sympathetic, parasympathetic, and enteric plexus. The enteric plexus has to do with a whole bunch of nerves that connect the stomach to the brain, and they call that the brain-stomach connection, and that's kind of a new area of interest to many pathologists. But sympathetic and parasympathetic are two very commonly well-studied divisions, where we have sympathetic is the fight or flight, and that mediates those sudden emergency responses that need to happen, while parasympathetic is going to be more the rest and digest division. So neurons are going to release chemicals to tell the next neuron to do something. So we start with some type of stimulation, some type of receptor, and that receptor senses a change in the environment. That change in the environment can be detected by causing a change in the neuron. And when that neuron changes enough to reach threshold is met, and it's going to travel down to the end of the neuron, release chemical messengers into the gap between the next neuron. Then the next neuron is going to receive those chemicals, and it's going to be woken up. And if it reaches threshold, then it's going to send an electrical nerve impulse, and ending with the release of chemicals from the end of the neuron. And this process is going to keep continuing. Conductivity, or excitability, so going through the definitions here, excitability is the ability to respond to stimuli. Conductivity is produce electrical signals that are conducted to other cells. Secretion, when the signal reaches the end of the axon, the neuron secretes a neurotransmitter that's going to then stimulate the next cell. So excitability, some type of stimulus happened and that neuron got excited. As soon as that initial neuron gets excited enough, it's going to reach a threshold point, and that's going to tip the scales and produce an electrical signal. And that electrical signal is going to travel down the end of that neuron. Once it reaches the end, you're going to have secretion. When the signal reaches that end, it's going to release a neurotransmitter that's going to then stimulate the next cell. Three functional classes of neurons: the sensory neuron, interneuron, and motor neuron. So we kind of already talked about this when we went through the divisions just now, but the sensory or afferent neurons detect stimuli and transmit information about them towards the CNS. So sensory are going to sense things in the environment, and then they're going to send that signal to the central nervous system. Inside of the central nervous system, we have interneurons, and interneurons receive signals from other neurons, process the information, and make resulting decisions. They lie entirely within the central nervous system, connecting motor and sensory pathways. Most common functional type, about 90% of all neurons. So again, central nervous system is brain and spinal cord. A lot of things are processed in the brain, but for reflexes, sometimes those reflexes just go through the spinal cord. They go in through the spinal cord quick, there's a response that comes out, and you have an immediate reaction. Other times, the brain needs to get involved immediately, and the brain is the one that makes those reactions. Again, we're going to learn more about those types of reactions when we get to next lecture. Motor or efferent neurons send signals out to muscles and gland cells, the effectors. So, if you hurt your finger, that finger is going to have sensory or afferent neurons conduct signals from the receptors to the central nervous system. Within the central nervous system, the interneurons are confined to the central nervous system. Those are going to make the decisions for what type of response we should have. The response is then determined and sent down a motor neuron to the effector. And the effector, the neurons conduct the signals from the central nervous system to effectors such as muscles and glands. Those are going to actually do the movement. So if you prick your finger, this signal is going to be sent down a sensory receptor to the interneuron, which is going to tell you to move your finger away from the source of the pain. So the motor neuron is going to tell your skeletal muscles to move, and then you're going to move. The structure of a neuron. So the principal components of a neuron, we have a cell body, an axon, and dendrites. And then we also have axon terminals. The cell body has all of the same types of organelles that you'd find in any normal cell. There's no centrioles because they can't undergo mitosis. As I talked about previously in the class, some cells just can't divide. And if they can't divide, then all you're going to see is the rest of the organelles. You're not going to see centrioles. So here's a picture of a neuron, and it's all nicely labeled here. So this is your typical neuron. You've got the cell body with all the typical organelles inside of it. This is where neurotransmitters are produced. We're going to learn a little bit about that later, but they're produced here, and eventually they're transported down to the end where they are released. The dendrites are on the surface of the cell body, and those are going to be able to detect changes in the environment. So whether it's chemicals that are going to bind to the dendrites, or if it's a sensory receptor, maybe just a physical touch, pain, some type of tissue damage, chemicals, signal from tissue damage, lots of different things can signal that something's changing in the environment. So dendrites are the receptive regions. They're going to pick up on those changes in the environment. Then you have this axon hillock. The axon hillock is where the signal accumulates. We'll learn more about what I mean by that once we get into the nerve impulse. The axon here transmits the impulse down to the axon terminals. On this axon, you're going to see Schwann cells and nodes of Ranvier, which are the spaces between the Schwann cells. When this nerve impulse goes down the axon, anywhere that it's covered by a Schwann cell, it's going to be transmitting the signal a lot faster. So it goes super fast, slows down, super fast, slows down, super fast, and that's kind of how it goes as it passes through to the end. And then these terminal branches and axon terminals, this is the secretory region. So once this neuron has been excited and that impulse reaches the end here, it's going to release neurotransmitters from those axon terminals. So just to give you a big picture view of what's actually going to happen during a nerve impulse. Let me see. We'll talk about it once we get to the conduction zones. Right now, we'll just keep it at the different structures that you see here. So myelin in both the peripheral nervous system and central nervous system, the myelin sheath is segmented. Many Schwann cells for the peripheral nervous system or oligodendrocytes for the central nervous system are needed to myelinate the axon. Each segment of the axon wrapped by a different glial cell. The myelin sheath gap, the node of Ranvier, is the gap between the segments. Then there's internodal segments, myelin-covered segments, the initial segment or bare section of an axon between the axon hillock and the first glial cell. Trigger zone, the axon hillock and that initial segment, plays an important role in initiating the nerve signal. The speed at which a nerve signal travels down an axon depends on two factors. One, the diameter. Larger axons have more surface area and conduct signals more rapidly. The presence or absence of myelin. Myelin speeds up signal conduction. So here's some examples provided. Small unmyelinated fibers, pretty slow. Small lightly myelinated fibers, a little bit faster. Large myelinated fibers, up to 120 meters per second. So those go pretty quick. So now we're going to give a big broad picture overview of the neural conduction, and then we'll go into the nerve impulse in a bit more detail. So here's the picture of the neuron. You already know the similar parts. So we've got the cell body here, then we've got dendrites, and it is important to mention that some textbooks refer to the cell body as the soma of the neuron. So if you ever see that term soma, that's referring to the cell body of the neuron. Then you have the axon hillock, the axon leading down to the axon terminals, and then those terminal knobs or synaptic knobs at the end. So, as we set the stage for a nerve impulse, we have to learn a little bit about electricity. In previous classes, you should have learned about intracellular and extracellular fluid compartments. Intracellular fluid compartments would be found inside of the cell. Extracellular fluid compartments are found outside of the cell. There's some major ions at play inside and outside of many cells. Sodium, Na plus sodium ion. Na is the chemical signal for sodium. Na plus, those types of ions are predominantly outside of the neuron, of every cell. They have a higher concentration outside of the cell in the extracellular fluid. Potassium, or K+, K is that symbol for potassium, K+ would be in higher concentration inside of the cell. Now, why aren't both of them in high concentration outside of the cell or inside of the cell? Because they're both positively charged, and positively charged things don't like to be by other positively charged things. We like charges repel. Opposite charges attract. So outside of the cell, surrounding the cell with those sodium ions, you're going to have a negatively charged ion. The one that's predominantly on the outside of the cell is going to be chloride, which kind of makes sense because sodium plus chlorine comes together to make sodium chloride, salt. So we've got sodium ions and chloride ions floating around in higher concentrations outside of the cell. Inside of the cell, to balance out those positively charged potassiums that are in there, we have negatively charged proteins. And those proteins are stuck on the inside. So they don't leave the cell, they're stuck on the inside of the cell. So now we're kind of establishing a concentration gradient. So we've got one type of ions, or one category kind of outside, one other type inside. Another thing to note is that we also have a sodium-potassium pump that continuously pumps to ensure that the sodium predominantly stays out and the potassium predominantly stays in. Now these membranes are super leaky to potassium, so potassium pretty much goes wherever it feels like it. So that sodium-potassium pump has to work 24/7 to make sure that the potassium stays inside the cell. The membrane's also kind of permeable to sodium, but not as much. So potassium can kind of flow in and out, sodium not so much. What we want to achieve is an overall negative charge inside and a positive charge outside, and that sets the stage for the resting membrane potential of -70. And we'll learn more about that. Now, what happens is when a dendrite receives a signal, or if a neurotransmitter binds to a dendrite, it's going to cause the opening of a chemically gated sodium channel. So now sodium is going to start slowly coming into this neuron. Now, if it's just one neurotransmitter that bound, sodium is slowly going to come in, and then it'll slowly reverse because we have those sodium-potassium pumps that are constantly working and constantly regenerating that resting membrane potential. Now imagine in this axon hillock here that there is an imaginary boat, and the cell body is a lake. And all these dendrites are like piers. So you're standing on a pier and you're throwing rocks into the lake. Now, if you throw one rock, you're going to start some waves, and those waves are going to come to the boat and they're going to start to rock the boat. So you throw another rock. And if you throw another rock, then you're going to make bigger waves and it's going to rock the boat even more. Now let's say many people from many of the different areas are all throwing rocks and all disrupting the water and making tons and tons and tons of big waves. Eventually, this boat is going to get to the point where it just tips over. Once that happens, if there's a person in that boat, that person has to make a decision to swim, and they have to swim all the way to the end because safety on shore is over here. You're not going to swim toward the people throwing rocks at you. Your boat tips, you're going to have to swim all the way to the other end. So that's kind of what happens here. We have this input zone where we are receiving signals either from the environment or from another neuron. Now, as the signals are received here, we're opening up ion channels, and that's going to cause a local depolarization of the membrane. In other words, the sodium that should have been outside is now going to start coming inside. Then you have the summation zone. Eventually, this is all going to reach a point where we have a signal accumulating here. And once the membrane potential, once it gets positive enough, once it reaches a certain threshold number, that's going to trigger an electrical impulse. Now, what happens once an electrical impulse is signaled, we no longer really care about what's happening up here, because whatever happened up here was enough to wake up or to tip over this boat, to wake up this nerve impulse. But in the conduction zone, it is an all or nothing response. So if this boat tips over, you're swimming. In other words, if that threshold membrane potential has been met in this summation zone, that electrical signal is going to be conducted. It changes from like a chemically gated channel system to an electrically gated channel system, and it's been induced now by this summation zone. That action potential is going to go all the way to the end, and it's going to go all continuously in one speed. Once it gets to the end, calcium is going to enter in there, and through a number of different processes depending on what type of neuron this is, predominantly going to be mediated by like calcium. These neurotransmitters are going to be released at the synaptic knobs. Now, when those neurotransmitters are released, they are free to go activate the next neuron. And the next neuron is then going to kind of start over here at the top. So the next neuron would then receive them. The signal would have to accumulate in here. Eventually, enough positive charge is inside of the membrane to cause this boat to tip over. The axon is going to fire and all the way to the end until we have that output zone reached, and you're going to have more neurotransmitters released. So hopefully that overview made a little bit sense. I can always go over it again in class or in tutorial session, but electrical potentials and currents. Neural communication is based on electrical potentials and currents. Electrical potential is the difference in concentration of charged particles between one point and another. Type of potential energy, it's measured in volts. Under some conditions, it can produce a current. Current is the flow of charged particles from one point to another. Example, a flashlight battery has a potential of 1.5 volts, can produce a current of electrons that lights the bulb. With a potential voltage, the battery is polarized. Living cells are also polarized. This is where we talk about the resting membrane potential. This is the charge difference across the plasma membrane. Typically, we're sitting at minus 70 millivolts in an unstimulating resting neuron. Negative value indicates more negatively charged particles on the inside of the membrane compared to the outside. So I kind of summarized this when I was talking about how the sodium ions are on the outside and there's some chloride ions on the outside. And you have those large negatively charged proteins inside and some positively charged potassium inside. Now, because potassium's kind of able to go in and out of the cell, and those negatively charged proteins, there's so many of them inside the cell, we have an overall negative charge inside of the cell and a positive charge outside of the cell. So we call this a resting membrane potential of about -70. That number is significant, so you should make a note of the -70 number. That's been pretty well documented that that is our resting membrane potential in an average resting neuron. Electrical currents in the body are created by flows of ions. So I already talked about sodium and potassium. At this point in your college career, you should know that Na refers to sodium and K refers to potassium. If not, definitely memorize it like as soon as possible, because those things aren't going to go away throughout your studies here. So make sure you know Na is sodium, K is potassium, and then there's a few others like Cl, chloride. So Cl by itself is chlorine, but Cl minus is chloride. Ca is calcium. So those are going to be really relevant when we talk about not only this nerve impulse, but then when we apply it toward the muscle contraction, you'll have to know what those ions are by name and by symbol. So there's different types of channels around the neuron. Leakage channels are exactly what they sound like. They're leaky. So substances can leak through them. But they're also specific, so that if you have a leakage channel, only one type of substance can leak through it. The best example in the neuron is the leakage potassium channels. They allow potassium through them. Gated means that the door is normally shut. So leakage, the door is always open. Anyone can come and go as they please. Gated, the door is normally shut. That door will only open for 2 reasons, typically. Or 3 reasons. We would have chemically gated, voltage gated, and mechanically gated. Chemically gated, also known as ligand gated, would be a chemical would have to bind to it in order to cause it to open. So a chemical would have to bind in order to open. So if we had a door on a bedroom and you wanted it open and you throw a bunch of acid at it, sure, eventually the acid will corrode through the door and it'll open. So that's one way of opening a door. Voltage gated. Same door in a bedroom in a house. Let's say it's a metal door and you electrocute it. So that's going to cause the door to fall off or open or explode or whatever. So voltage gated only opens in response to a change in voltage. Mechanically gated would only open in response to a physical movement or a physical change in protein structure. The opening and closing of doors may or may not require energy, but what does require energy is that sodium-potassium pump, which we'll learn about as we go through here. That sodium-potassium pump is kind of like a gated, not gated channel, a facilitated protein channel, and that is going to, through active transport, cause sodium to be pumped out of the cell and potassium to be pumped in the cell to maintain that -70 resting membrane potential. So the resting membrane potential, the ionic basis of the resting membrane potential, ions are unequally distributed between extracellular fluid and intracellular fluid. The resting membrane potential results from a combined effect of the diffusion of ions down their concentration gradients through the membrane, selective permeability of the membrane, allowing some ions to pass more easily than the others, electrical attraction of the cations and anions to each other. Potassium has the greatest influence on the resting membrane potential. It's more concentrated in the intracellular fluid than the extracellular fluid. The membrane is leaky to potassium more so than other ions. If potassium leaks out, the inside becomes negative and pulls potassium back in. Equilibrium is met when there is no net change in membrane potential. Potassium in equals potassium out. Sodium also influences resting membrane potential. Sodium is more in the extracellular fluid than intracellular fluid. The membrane is less permeable to sodium than potassium. Relatively small amount of change on the resting membrane potential. The sodium-potassium pump requires ATP, must work constantly to account for the leaky channels, pumps sodium out and potassium in. The sodium-potassium pump accounts for 70% of the energy requirement of the nervous system as a whole. So that's pretty significant. So because the sodium-potassium pump requires energy, if we die, we're no longer bringing in oxygen. And if we're not bringing in oxygen, we're not able to produce energy. And without energy, we can't have any nerve impulses. So this is why neurons stop firing. So here is a diagram that you can review. Some people find these types of diagrams useful. You can see where the sodium-potassium concentrations are. You'll notice that both sodium and potassium are on both sides of the membrane. But if you look at the concentrations, there's like 145 versus 12. So you can see it's much higher on the outside of the membrane. Where if you have potassium, it's the opposite. It's much higher on the inside of the membrane. Inside, you have these large anions that can't escape. That's those negatively charged proteins that are stuck. And that's what contributes to the overall negativity inside of the cell. Local potentials can be graded, vary in magnitude with stimulus strength. Stronger stimuli open more sodium channels and they stay open longer. Decremental, they get weaker the farther away they spread from the point of stimulation. So decremental would be kind of like throwing the rocks in the water. If you have stood at the end of a pier and thrown a rock in the water, you'll notice that the waves are really close together where the rock just hit. But as it gets farther and farther away, the waves get spread out more and more and more until the point where they don't even affect the water around it anymore. So that would be like a decremental example. Reversible. If the stimulation ceases, the membrane voltage quickly returns to normal resting potential. They can be either excitatory or inhibitory. Depolarization is excitatory. It makes a neuron more likely to fire an action potential. Hyperpolarization, though, membrane becomes more negative, is inhibitory. It makes a neuron less likely to produce an action potential. An action potential is sometimes also exchanged for nerve impulse. But that action potential is talking about once it reaches that axon hillock and it triggers the electrical conduction down the axon, that's what we're referring to as like the action potential. It's firing down that axon. So the excitation of a neuron by chemical stimulus. So here you've got dendrites, and if we blow up a picture of the membrane by the dendrites, we've got the resting membrane potential where we've got a bunch of sodium outside, not a lot of sodium inside. The outside of the membrane is overall positive, and the inside of the membrane is overall negative. So we have a -70 resting membrane potential. If there's a stimulus that binds to the dendrites, like let's say another neuron released some neurotransmitter. Well, now the neurotransmitter is going to go across this gap, and then it's going to bind to a sodium channel. This is known as a ligand-gated sodium channel, which I always was taught chemically gated sodium channel. Chemically gated just means that if it needs something to bind to it, like a chemical to bind to it in order for it to open. So once that specific chemical binds to the channel opens and a lot of potassium comes rushing in. That influx of potassium causes a positive charge to come inside of the membrane. That leads to depolarization. So polarization means you have opposite charges on either side of the membrane. Depolarization means that you are reducing the opposite charges. And in this case, you're making the inside more positive. Action potential, rapid up and down change in voltage produced by the coordinating opening and closing of voltage-gated ion channels. It only occurs where there's high enough density of voltage-gated ion channels, the trigger zone of the axon. If excitatory local potential reaches the trigger zone and it's still strong enough, it opens the enough voltage-gated sodium channels to generate an action potential. So this is where that diagram where I showed you the input zone, and you had to kind of build up waves, and all those waves had to get to that axon hillock with enough strength to tip over the boat. And why do they have to get to the axon hillock? Because that's the only place where there is a high enough density of voltage-gated ion channels. That's that trigger zone of the axon. Once those voltage-gated ion channels open, though, it's like a domino effect. As soon as you tip the first domino over, there's no stopping it. It is going to go all the way to the end continuously until it reaches those axon terminals. So the steps of an action potential. Local potential spreads to the axon hillock. Voltage at the axon hillock must reach threshold. The minimum voltage to open a voltage-gated channel is around minus 55 millivolts. I'm not going to make you memorize every number, but you do have to know the minus 70 of the resting membrane potential. You don't need to know the minus 55. Voltage-gated sodium channels open quickly. Voltage-gated potassium channels open more slowly. Sodium enters, depolarizing the membrane further. The sodium channels become inactivated and begin to close, and the influx of sodium stops. So here you can see they're both closed, but if it reaches threshold, threshold means that there's enough of the charge difference that met these voltage-gated ion channels. The sodium ion channels in the axon are going to open. Now the potassium ones are going to open slightly, but because these sodium ones open so much, that's going to cause the initiation of that action potential. And that's what is discussed here. The voltage-gated potassium channels are then going to fully open, and potassium flows out of the cell, and the membrane becomes more negative again. So repolarization happens. So you have like 3 sets of dominoes right next to each other. The first set of dominoes you're going to knock over, and it's going to travel down the axon, opening up those voltage-gated sodium channels. But before it even reaches the end, the next row of dominoes that's standing right next to that initial row is going to be pushed, and those potassium channels are going to really open up, and potassium is going to start to go out of the cell. So you're going to have a wave of depolarization and then a wave of repolarization. Then what happens is that third row of dominoes causes even more potassium channels to open, which causes even more potassium to exit. So now you've got extra potassium leaving the cell, and that's hyperpolarization. When a cell has been hyperpolarized, it is really difficult to overcome that. So we will send the nerve impulse all the way to the end through depolarization, then repolarization, then hyperpolarization. And you can again picture those 3 rows of dominoes kind of going all the way down to the end of the axon. And then there's like some resetting that has to occur before this action potential can happen again. And here you can see the relative voltages. So during resting membrane potential, we're at minus 70. Once we reach threshold, which varies, but they're depicting it at minus 55, we're going to start to have depolarization. Then through repolarization, we are eventually going to get back down to threshold, and then through hyperpolarization, we're going to get even lower than what resting membrane potential would have been. It's through this gap that prevents us from continuously firing nerve impulses down the same neuron. Sometimes, like let's say you prick your finger on a needle. Well, you're going to have one nerve impulse taking that information up to the central nervous system. Your central nervous system is going to process it real quick, send it down to the motor neurons, and your motor neurons are going to have a reflex that jerks your finger away from the source of the pain. Then, after that initial reflex, then finally you start to feel the pain. So now you have a sensory receptor coming in from the pain receptors, and your brain then processes the pain. And then your brain kind of evaluates what just happens. And then you're like, oh, yeah, that was dumb. That really hurt. And you process it a little bit more. Now, that pain is going to continue. And it may even feel like the pain is throbbing. And that's because the pain and processing in the brain is enough to overcome that hyperpolarization and send down another action potential. But your initial action potential was real quick, one time, and it wasn't a big enough deal to overcome hyperpolarization. A nerve impulse is a nerve impulse no matter what. Once you trigger that axon hillock and you trigger that action potential to go to the end, it's going to go continuously in one speed, just like knocking over dominoes. So how does your brain determine if you have pricked your finger on something or blown your arm off? So very different things, very different reactions. Well, you determine it based on the number of nerve impulses. So pricking your finger is going to stimulate a small number of neurons. It's going to hurt. You're going to notice it. You're going to jerk your finger away. It's going to be painful for a period of time, but you quickly get over it. Now, if your arm blows off, obviously many neurons are going to be stimulated, and you're going to have signal after signal after signal being sent down their action potentials. So that sends a big sign to the brain that something major is going on in the body. So it's not like the speed of the nerve impulse that tells your brain it's significant, because if it's a neuron and it's one type of neuron, the signal is going to travel down that neuron the same speed no matter what happens. It's the number of action potentials that are allowed to go down. And it's this hyperpolarization that's difficult to overcome. You actually have to get back up to resting membrane potential before you can even think about starting another action potential in most cases. And also, if you remember, sodium went in the cell, potassium went out of the cell. Now that's all upside down. We have to reset the membrane and get these ions back to where they should be. So here's that picture I showed before. So normally they're closed. If we have the voltage met, if a threshold has been met, then the sodium channels are going to open, potassium's going to open slightly. Then we have sodium channels close and potassium's going to open. Then you have a rush of potassium ions leaving the cell. And then you have that hyperpolarization happen where you're going to keep having that potassium being pumped out of the cell. Characteristics of the action potentials, the all or none law. Once threshold is reached, the neuron fires up to maximum voltage. If threshold is not reached, it does not fire. You either tip the boat over or you don't. So once that boat's tipped over, you have no choice but to swim. Non-decremental, it does not get weaker with distance. So in the action potential, like unlike the cell body, so we already talked about the cell body, we had some depolarization, but from the source, it gets weaker and can even be stopped if it's not strong enough to tip the boat over. Once we reach the axon though, it's an all or nothing, and it's going to continue or go down continuously. And it cannot be stopped. So irreversible, it cannot be stopped. It's going to go all the way to the end no matter what. Just like tipping over that domino. The dominoes in a row, they're all going to fall over all the way to the end at a continuous speed. The refractory period is a period of resistance to stimulation, and it has two phases. The absolute refractory period, no stimulus of any strength will trigger another action potential, is caused by the inactivation of voltage-gated sodium channels. Then you have the relative refractory period, which is an unusually strong stimulus is needed to trigger a new action potential. This is during hyperpolarization. A larger depolarization is required to reach threshold. So that's where, like, if you pricked your finger versus your arm being blown off is going to be able to overcome this hyperpolarization. So here you can see the resting membrane potential. Then you have this absolute refractory period that no matter what, nothing can overcome it. And then you have the relative refractory period where the membrane's kind of resetting and we're trying to get back up to normal. So the continuous conduction of a nerve signal in an unmyelinated axon. So here you can see the action potential, the refractory membrane, and a new excitable membrane. And those will follow down after each other. So a very strong impulse would be able to start an action potential earlier. And the brain receives the number of action potentials and can determine the severity based on the number of action potentials. Myelinated axons and saltatory conduction. So action potentials can only be generated at the nodes where voltage-gated ion channels are concentrated. Electrical signal must spread passively between nodes. The signal passes very quickly, but strength decreases, similar to a local potential. When the signal reaches the next node, it's still strong enough to depolarize the membrane to threshold. Voltage-gated sodium channels open and a new full-strength action potential occurs. Action potential seems to jump from one node to the other. It moves faster through the insulated segments covered with myelin and slows down when it reaches the bare axon of the nodes. So here you can kind of see if they're insulated, the ions don't escape. It's in the nodes that the ions are allowed to kind of float farther away, and that causes disruption in the signal. All in all, the action potential is able to continue all the way down to the end. Synapse, the point where an axon terminals meets the next cell or another neuron. For neuron-to-neuron synapses, the action potential arrives at the axon of the presynaptic neuron. Here, pre and post are important. Pre means before it hits the gap, the synapse, the space between you and the next, or one neuron and the next neuron. Presynaptic neuron releases a neurotransmitter, and the postsynaptic neuron responds to it. So let's say that you and your friend live next door to each other, and the phone lines are down, and you can't use cell phones, all cellular signals down. And you want to get your neighbor's attention because you just got some news that something bad's happening and you need to get a hold of them and you need to let them know and relay the signal to them. You can't go outside. Something's going on outside. So you're in your house and you look through your window and you start throwing things at your neighbor's window. So you throw a rock, they don't hear it. You throw a couple rocks back to back, they still don't hear it. So then you decide to throw something big, like let's say you throw a boulder at their house. Well, now they're probably going to hear it because it made a super loud noise. So in our house, we are the presynaptic neuron. The rocks that we're throwing are going to be neurotransmitters. And the number of neurotransmitters that we are going to throw is going to be dependent on the number of action potentials that actually make it to the end of us. So the severity of the news that we hear, that's going to determine our response. So I'm going to throw like maybe one rock if I want to get their attention because I'm bored, maybe like 50 rocks if I want to get their attention because their house is on fire. So presynaptic neuron, that's me. Postsynaptic neuron, that would be the neighbor's house. And I'm going to be throwing rocks to try to get their attention. That's literally how neurons communicate with one another. One neuron gets excited, and then it sends a signal. And then it's going to release chemicals in accordance with how many action potentials actually make it down to the end. Those chemicals are going to be released, and they're going to travel across the synapse and bind to the dendrites of the next neuron, the postsynaptic neuron. That postsynaptic neuron is then going to start the depolarization process. And then their cell body is going to depolarize to the point where if threshold is met, they're going to fire their own action potential. And that's kind of how that signal is carried from neuron to neuron. And this just shows that in a diagram. There's different types of connections that can exist. You can see axodendritic, axosomatic, so that's the axon to the cell body or the axon to the dendrites. We can also have the axon to another axon and stimulate the end of the axon. And a lot of these different connections are found in various regions of the body for different types of reasons. Our most common one, though, would be the cell body axon. We've got axon terminals, which would then synapse with the next dendrites. That synaptic cleft, that's just the name of the gap between the presynaptic neuron and postsynaptic neuron, between my house and my neighbor's house. Typically only 20 micrometers wide. Each neuron has cell adhesion molecules, or CAMs, reaching into the cleft. CAMs link two neurons together. Axon terminal presynaptic neuron contains synaptic vesicles containing the neurotransmitters. Many vesicles are docked on release sites on the plasma membrane that are ready to release neurotransmitters. The postsynaptic neuron membrane contains postsynaptic density of neurotransmitter receptors and ion channels. Ligand-gated ion channels open when neurotransmitters bind to them. So here you can see axon of presynaptic neuron, axon terminal, and the cell body of the postsynaptic neuron. And here's another diagram that kind of shows you the different parts here. Sometimes students like looking at these diagrams. I'm never going to pull up a diagram and make you label the different parts that you're seeing on these. So chemical synapse, so as soon as the electrical signal gets to the axon terminals, we already have vesicles containing neurotransmitters waiting here. And they're waiting to be released in that synaptic gap. When we have the postsynaptic neuron, we've got all these receptors where if one of those neurotransmitters were to bind to the receptor, it then opens this gate. So this is a ligand-gated channel, and that's going to stimulate depolarization of the next neuron. There's more than 100 neurotransmitters that have been identified, and most of them fall into these major categories. Acetylcholine, formed from acetic acid and choline, that's going to stimulate our muscles. So we're going to learn a lot about that when we talk about how muscles are stimulated. Amino acids, including glycine, glutamate, aspartate, and gamma-aminobutyric acid, or GABA. Monoamines, synthesized from amino acids by removal of the carboxyl group, but retain the amino group. Examples, the catecholamines, epinephrine, norepinephrine, and dopamine, also serotonin and histamine. Purines, adenosine, or ATP. Gases like nitric oxide and carbon monoxide, those are synthesized as needed rather than stored in vesicles. Neuropeptides, chains of 2 to 40 amino acids, cholecystokinin, and endorphins. Stored in large secretory granules, some also function as hormones or neuromodulators. So where are neurotransmitters produced? So as we talked about the process of protein production, neurotransmitters are going to be made in the cell body. The genetic material codes for those substances, and sometimes if they're lipid-based, they can be produced in the Golgi apparatus. Then they're going to be packaged in vesicles. They attach to the cytoskeleton, and then they're going to be transported down the axon all the way to the end to the axon terminals. Here, they're going to be snagged and stored. When an action potential enters, then we have a series of events that takes place to release those neurotransmitters from the end. So electrical versus chemical synapse. So sometimes instead of me throwing rocks at my neighbor's house, our houses are connected directly. So anything that happens in my house, I can immediately knock on the door and pass the signal on to the next house. That's kind of like gap junctions. And gap junctions are found in heart cells because we have to be able to, once that signal is triggered that we want the electrical conduction system to start in the heart, we want that to transmit to every single heart cell that it possibly can on the top part of the heart, and then kind of regroups at the next segment of that conduction system. And we want the signal to transfer to every single heart cell in the bottom half of the heart. So those electrical type signals, those would have to happen at gap junctions, and that's a very fast and rapid and direct process. Now the good thing to that is it can happen fast. The bad thing is if our entire body was wired that way, any little tiny nerve impulse that was stimulated would automatically cause massive reactions in our bodies because they would for sure stimulate the next neuron and the next neuron and the next neuron and the next neuron. Sometimes our brains don't need to get involved. If we get a tiny injury and it's something that like maybe just the surface of the skin got cut a little bit, but it's not something that any pain receptors were involved because all it did was cut through some dead skin cells, our brain doesn't need to get involved in something like that. So we don't necessarily want that nerve impulse making its way to the brain. If we had gap junctions in every single one of our neuron connections, then those signals would automatically be transferred to the brain, or they would automatically be transferred along the next step in the pathway, and that's something that would cause overreactions in our body. So it's kind of nice that we have this synaptic gap. So if we have to release the neurotransmitters, sometimes that can kind of be a quality control mechanism of making sure that we're only continuing the signal if the signal is worth continuing, or if the signal is strong enough where it merits that continuation. In other words, if we end up not getting multiple nerve impulses and we only release one neurotransmitter, that one neurotransmitter would bind, cause a local depolarization, but it would never get to the point where it would build up enough to have a change inside the cell. If we were all functioning in gap junctions, like what happens in our hearts, doesn't matter. You could have one electrical signal get there and it's going to fire to the next one no matter what. Synapses are variable in their modes of action. Some neurotransmitters are excitatory, others inhibitory, and sometimes a neurotransmitter's effects differs depending on the type of receptor on the postsynaptic cell. Some receptors are ligand-gated ion channels, others act through intracellular secondary messengers. Examples of three kinds of synapses: excitatory cholinergic synapse, inhibitory GABAergic synapse, or excitatory adrenergic synapse. In an excitatory cholinergic synapse, acetylcholine is the neurotransmitter. So an action potential depolarizes the axon terminal, and that opens voltage-gated calcium channels. Calcium enters and triggers exocytosis of acetylcholine. Exocytosis is when you release large amounts of substances from the cell. So basically the vesicle is going to fuse with the cell membrane and it's just going to release those acetylcholine neurotransmitters out into that synaptic gap. Acetylcholine is going to diffuse across the gap or the cleft and bind to the postsynaptic receptors. Acetylcholine receptors are ligand-gated ion channels that open and allow sodium and potassium across the membrane. Entry of sodium causes depolarization. Postsynaptic potential: if it's strong enough, the depolarization spreads to the trigger zone, raises the membrane potential up to threshold, and then another action potential is triggered. So this shows a picture of that. So you can see that calcium, once the action potential reaches the end, calcium enters into the cell, which causes this vesicle to actually fuse with the cell membrane, releasing the acetylcholine. Acetylcholine is going to go across the synaptic gap, and if the receptors are present, the receptors are going to be there. Acetylcholine is going to bind and then cause local depolarization. Here's another diagram of the same thing. So we've got that action potential coming down here to the end. These vesicles already are here. They've already been created, and they're just waiting until an action potential hits. When the action potential does hit, voltage-gated calcium channels are going to open. And you see Ca++, it's because it's 2+. That's the ionic form of calcium is Ca2+. Sometimes it's written as +2. But here you can see calcium mediates the fusion of these vesicles with the outside membrane, and that causes the release of these neurotransmitters across the synapse. An inhibitory GABAergic synapse, gamma aminobutyric acid is the neurotransmitter. Steps in synaptic transmission, an action potential releases GABA. GABA receptors are chloride channels. Chloride entry hyperpolarizes the postsynaptic membrane, and the postsynaptic neuron is inhibited, less likely to fire an action potential. Adrenergic, norepinephrine is the neurotransmitter. Norepinephrine is also called noradrenaline. Monoamines and neuropeptides bind to G protein-coupled receptors on the postsynaptic membrane. It activates a second messenger system, such as cyclic AMP or cAMP. It's slower to respond than cholinergic and GABAergic synapses, and has advantage of enzyme amplification. A single molecule of norepinephrine produces vast numbers of product molecules in the cell. And here's a picture of that secondary messenger system that would have downstream effects. Cessation of the signal. So at some point we got to stop the signal. It's important not only to stimulate it, but also to turn it off. The neurotransmitter stays bound to the receptor for about 1 millisecond. If the presynaptic cell continues to release neurotransmitter, one molecule is quickly replaced by another, and the postsynaptic cell continues to be stimulated. To end the signal, the presynaptic cell stops releasing neurotransmitter, and the neurotransmitters that are already in the synapse have to be cleared in various ways. The main ways are going to be neurotransmitter degradation. There's enzymes in the synaptic cleft that break down the neurotransmitter. An example would be acetylcholinesterase breaks down acetylcholine into choline and acetate. Reuptake, sometimes neurotransmitters or its breakdown products are reabsorbed into the axon terminal. For example, choline from acetylcholine is recycled to make new acetylcholine. Amino acids and monoamines are also reabsorbed, degraded in axon terminal by the enzyme monoamine oxidase, the target of some antidepressant drugs. Diffusion, the neurotransmitter or its breakdown products simply are diffused away from the synapse into nearby extracellular fluid. So here you can see those processes happening. Reuptake, it's reabsorbed by the presynaptic axon. Enzyme degradation, enzymes just break it apart. For example, acetylcholinesterase is an enzyme that can break down acetylcholine. And diffusion, sometimes these things just diffuse away. Neuromodulators, so chemicals secreted by neurons that have long-term effects on groups of neurons. They may alter the rate of neurotransmitter synthesis, release, reuptake, or breakdown. They may adjust the sensitivity of postsynaptic membrane. Nitric oxide is a simple neuromodulator, or not modulator, gas that enters the postsynaptic cells and activates second messenger pathways, for example, relaxing smooth muscle. Neuropeptides are chains of amino acids that can act as neuromodulators. Enkephalins and endorphins are neuropeptides that inhibit pain signals in the central nervous system. Neural integration, the ability to process, store, and recall information and use it to make decisions. Chemical synapses allow for decision making, Brain cells are incredibly well connected, allowing for complex integration. There's pyramidal cells of the cerebral cortex have about 40,000 contacts with other neurons. The trade-off, though, chemical transmission, involves a synaptic delay that makes information travel slower than if there was no synapse. There's two types of postsynaptic potentials that are produced by neurotransmitters, an excitatory and an inhibitory, and these are abbreviated EPSP for excitatory postsynaptic potential or IPSP for inhibitory postsynaptic potential. In an EPSP, the voltage change from the resting membrane potential toward threshold. An EPSP usually results from sodium flowing into the cell. IPSP, voltage becomes more negative than it is at rest. An IPSP can result from chloride entry or potassium exit from the cell. Different neurotransmitters cause different types of postsynaptic potentials. Glutamate and aspartate produces EPSPs in brain cells. Glycine and GABA produce IPSPs. A neurotransmitter might excite some cells and inhibit others, depending on the type of receptors in the postsynaptic membrane. Acetylcholine and norepinephrine work in this way. Acetylcholine excites skeletal muscle but inhibits cardiac muscle due to the expression of different types of acetylcholine receptors on the different types of muscle cells. Summation is the process of adding up postsynaptic potentials and responding to their net effect, and this occurs in the trigger zone. Some incoming nerve fibers may produce EPSPs while others produce IPSPs. A neuron's response depends on whether or not net input is excitatory or inhibitory. The balance between EPSPs and IPSPs enables the nervous system to make decisions. Two ways EPSPs can be added to reach threshold. Temporal summation. A single synapse generates EPSP so quickly that each is generated before the previous one fades. It allows EPSPs to add up over time to a threshold voltage that triggers an action potential. Spatial summation, EPSPs from several different synapses add up to threshold at an axon hillock. Simultaneous input from multiple presynaptic neurons is required for the postsynaptic neuron to fire. An example of facilitation, a process in which one neuron enhances the effect of another. And here's a diagram. You can go ahead and pause the video and review this diagram. Neurons can work in groups to modify each other's actions. Presynaptic facilitation occurs when one presynaptic neuron enhances another one, increases necessary synaptic transmission. Example, facilitating neuron, called F in the figure, releases serotonin, which makes voltage-gated calcium channels in the axon terminals and stay open longer. Here you can see presynaptic facilitation in action, and there's a couple diagrams that you can pause the video and read through. Presynaptic inhibition occurs when one presynaptic neuron suppresses another one. It reduces or halts unwanted synaptic transmission. Example would be inhibiting neuron releases GABA, which prevents voltage-gated calcium channels in the axon terminal from opening, so little or no neurotransmitter is released. And here's a diagram of that. You can go ahead and pause the video and review what's going on in this diagram. And we come to memory. So short-term memory retrieval is rapid and involves recalling information held temporarily in the brain, primarily in the prefrontal cortex. It relies on active neural circuits and fades quickly unless rehearsed or transferred to long-term memory. Long-term memory retrieval involves accessing stored information from the hippocampus and cerebral cortex, often requiring cues or associations. Retrieval strength depends on factors like repetition, emotional significance, and neural connections, with stronger memories being recalled more easily.